In June of 2025, in laboratories in Beijing and institutions across the world, something extraordinary happened. A mystery that had haunted paleoanthropology for 15 years suddenly resolved itself. And when it did, it resolved not in the way that anyone expected. A skull that had been hidden in a well for nearly 90 years, a skull that had sparked scientific controversy just 4 years. Earlier, a skull that the world called Dragon Man turned out to be something far more profound than a new species. It turned out to be a face, the first face anyone had ever seen of the Denisovans, the most enigmatic human cousins in our evolutionary lineage. To understand what this means, you first have to understand what we did not know. And what we did not know is almost everything about them. 15 years before Dragon Man was identified, before anyone was even looking in this direction, a young girl's finger bone was discovered in a Russian cave. The cave was called Denisova Cave, nestled in the Altai Mountains of Siberia. And when geneticists sequenced the DNA from that tiny fragment of bone, they discovered something that should have been impossible. They discovered a human species that no one knew existed, not Neanderthals, not Homo sapiens, something else entirely. A third branch of the human family tree, living at the same time as both Neanderthals and modern humans, sharing ancestry with both, yet distinct from both. They called them Denisovans, after the cave where that finger was found. But here was the problem that defined the next 15 years of research. The Denisovans, unlike Neanderthals or Homo sapiens, left almost no fossil record. We knew they existed because their DNA survived in the bones of other humans, in Neanderthals, in modern Melanesians, in Tibetan populations, in people scattered across Southeast Asia and Oceania. We could read their genes in the genetics of living humans, trace their legacy through millennia, understand how much of modern human DNA carried the fingerprints of Denisovan ancestry. We knew their code. We just did not know their face. We did not know what they looked like. We had never held in our hands a skull, or even most of a skull that could tell us. The scientific world was therefore left in an extraordinary position. We had the genetics of a vanished species, but not the anatomy. We had the ghost, but not the body that had cast it. Dozens of fragmentary remains had been attributed to Denisovans, jaw, fragments, isolated teeth, scattered bone pieces from caves across Asia, but nothing complete. Nothing that would allow anyone to say with certainty what a Denisovan actually was. And then, in 1933, in the city of Harbin in northeastern China, a man was digging. He was building a foundation for a bridge across the Songhua River in a region that was occupied by foreign powers when his shovel struck something hard. He pulled from the earth a nearly complete human skull. It was massive. Heavy brow ridges, a flat face, a wide nose, prominent chin, a face that looked at once human and not quite human. It was extraordinary. It was also terrifying because he knew what would happen if he reported it. The skull would be taken from him. So he did what countless people in archaeology have done across time and space. He concealed it. He took it home. He hid it in a well at the bottom of a shaft, where it sat in darkness for 85 years. It sat there through wars and revolutions, through the entire history of modern genetics, while scientists around the world were asking themselves what a Denisovan looked like, and this man had the answer buried beneath his own house. The skull remained hidden until 2018, when the man finally came clean to his family, and they retrieved it and donated it to science. A team of researchers examined it. They looked at its morphology, its distinctive shape, the features that made it different from any other known hominin species, and they named it Homo longi, which translates as Dragon Man, after the Longjiang Dragon River of the region. They published their findings in 2021 and argued that they had discovered a new species entirely. Not a Denisovan, not a Neanderthal, something that deserved its own place on the family tree. The scientific community was skeptical. One more branch on an already complicated tree, one more hominin species that existed only in China. It did not quite fit the narrative. The majority of researchers were not convinced, but the skull sat in the museum anyway, still. Mysterious, still arguing for its place in human evolutionary history. The controversy was not resolved. The mystery simply sat. And then Qiaomei Fu, a geneticist at the Institute of Vertebrate Paleontology and Paleoanthropology in Beijing, made a phone call. She wanted to try to extract DNA from the skull. She wanted to know what the molecular evidence would say. The team that had discovered the skull agreed to allow her to work with it. And what happened next was a masterclass in methodological innovation forced by circumstance. Ordinarily, when paleogeneticists want to extract ancient DNA from human fossils, they target the petrous bone, a dense piece of bone at the base of the skull. The petrous bone is special. It is so dense, so hard, that it preserves DNA longer and more completely than other bones. It is the default target for ancient DNA extraction. But there was a problem in this case. The petrous bone had been removed from the Harbin skull decades earlier, when it was in collectors' hands. It was simply gone. Jiao Meifu faced a choice. She could give up, or she could gamble. She could try something that had only rarely been attempted at this scale, extracting DNA from dental calculus. Dental calculus is tartar, the hard mineral deposit that builds up on teeth over a lifetime, entombing bacteria and organic material in a rock-hard matrix. Normally, it would not be anyone's first choice for ancient DNA: extraction. But Fu realized something that few people had fully grasped. Dental calculus is also a time capsule. It is a sealed environment, protected from contamination by the very hardness that makes it calculus in the first place. And if you can crack into it, if you can access what is protected inside, you might find genetic material that has been preserved for tens of thousands of years. From a single tooth, from 0.3 milligrams of dental calculus, Jiao Meifu extracted DNA. And when she sequenced it, the mystery dissolved. The mitochondrial DNA, the DNA that passes from mother to child unchanged for thousands of generations, matched the Denisovans. It was not a perfect match. It was early in the Denisovan lineage, suggesting an individual who lived before the Denisovans, that were later found in Siberia. But it was unmistakably Denisovan. The Dragon Man. The Dragon Man was simply the first face that anyone had ever seen of the Denisovans, and what a face it was. But the DNA told only part of the story. A parallel team analyzed proteins extracted from the same samples. They used mass spectrometry to identify protein fragments. Preserved in the dental calculus and the petrous bone, the proteins revealed something even more striking than the DNA alone could convey. This individual did not just carry Denisovan DNA. The protein evidence showed 27 genetic variants that were found in the 7 known Denisovan individuals and nowhere else. The fingerprint was so complete, so unmistakable, that the skull that had been buried in a well for 90 years was the first neither Neanderthals nor Homo sapiens were built. The brain case was large, suggesting a large brain, but shaped differently from modern human brains. The face was broad and flat, adapted perhaps for processing tougher foods, for surviving in environments that required different mechanisms than those that had shaped modern humans. The individual appeared to be male, adult, somewhere between 30 and 40 years old when he died. He died 146,000 years ago, give or take 10,000 years, at a time when Neanderthals ranged across Europe and Western Asia, when modern humans were still confined to Africa, when this Denisovan roamed the forests and steppes of northeastern China, fully adapted to his world, fully successful in his niche, with no idea that his entire species would vanish from the earth within 100,000 years, leaving behind only genetic ghosts in the DNA of modern humans. Skull. It was that this discovery opened up an entirely new avenue of understanding how these people lived, where they traveled, and what they looked like. The morphology of the skull told stories that genetics alone could never convey. The robustness suggested a lifestyle of physical exertion. The teeth suggested a diet that included tough, fibrous plants. The bone structure suggested adaptations to a cold climate, perhaps. Every curve and ridge of the Harbin skull became a window into the life of a species that had been known only through fragments. And the discovery did something else. It changed the way that paleogeneticists thought about extracting DNA from ancient fossils. If DNA could be preserved in dental calculus, then specimens that had been declared too degraded for genetic analysis suddenly became viable again. Hundreds of fossils stored in museums around the world, specimens where the best bone samples had been removed or degraded, could now potentially yield genetic information. It was not just that we had finally put a face on the Denisovans. It was that we had developed a new tool for putting faces on all sorts of ancient mysteries. The implications rippled outward from that laboratory in Beijing. Researchers realized that they could revisit specimens that had been assigned to other species and reevaluate them. Already, a jaw discovered in the Penghu Channel between China and Taiwan was re-examined and found to carry Denisovan markers. Bone samples from caves in Tibet and other regions of East Asia were subjected to new analysis. Suddenly, East Asia was becoming a landscape covered with Denisovan fossils. They had been there all along. We just had not known how to recognize them. The broader picture that emerged was of a Denisovan population that had ranged far more widely than anyone had previously understood. The Siberian Denisovans, known from the finger bone and a few other fragments from Denisova Cave, represented one lineage. The Harbin individual represented an earlier, more eastern population. The dating suggested that Denisovans had occupied a vast range from Siberia to northeast China, probably further south as well, through the period from roughly 217,000 years ago to perhaps 106,000 years ago, overlapping geographically and temporally with both Neanderthals to the west and early Homo sapiens to the south. Picture this landscape. Imagine walking across Northeast Asia 146,000 years ago. You might encounter a Denisovan hunting party. Move west, travel toward the steppes and mountains, and you might encounter a Neanderthal group adapted to cold and tough with their own mastery of their environment. Move south toward Africa and beyond, and you might encounter early Homo sapiens, not yet modern in every respect, but recognizably our species, just beginning the expansionist journey that would eventually colonize the entire world. 3 species of human cousins, occupying the same vast continent, each successful in their own regions, each adapted to their own niches, each living out their lives with no idea that only one of them would ultimately survive. But they were not entirely separate. The DNA evidence that had been accumulating over 15 years showed that these 3 species had encountered each other. They had interbred. Modern humans carry between 3 and 5% Neanderthal DNA. Modern humans also carry Denisovan DNA, though the percentage varies dramatically by geography. Melanesian populations carry roughly 4 to 6% Denisovan DNA. Tibetan populations carry Denisovan genes related to altitude adaptation. Even in European populations, small traces of Denisovan genetic material can be found, suggesting encounters in gene flow that must have occurred in the distant past. This was one of the great secrets that ancient DNA had uncovered. We were not alone. We did not sweep across the globe and eliminate our competitors. We encountered them. We mixed with them. We absorbed some of them into our genetic fabric. Parts of them live on in us. The Harbin skull gave a physical, morphological reality to what had been purely genetic. This was what the Denisovans looked like. This was the species that had contributed genes to modern human populations. This was the face of a human cousin from 100,000 years ago. And now that we had that face, we could look at other fossils with new eyes. We could look at the jaw from Taiwan and see a Denisovan face in profile. We could look at other skulls from East Asia and ask whether they, too, belonged to this lineage. The story of the Harbin skull was also a story about how science makes progress. It was a story about a man who broke the law to save an artifact from destruction, who held onto it for most of his life, who finally came clean so that the world could know what he had known for so long. It was a story about scientists working across international lines, Chinese scientists and international collaborators, asking questions that crossed political and cultural boundaries. It was a story about methodological innovation forced by circumstances. Xiao Meifu could not extract DNA the usual way, so she tried something different, and that something different opened up new possibilities for the entire field. It was also a story that highlighted the power and the limits of genetics. The mitochondrial DNA told us who this individual was. The protein evidence confirmed it, but the DNA alone could never have told us what the individual looked like. The DNA alone could never have shown us the massive brow ridge, the flat face, the wide nose, the unusual brain case shape. Those features required morphological analysis. They required looking at the skull with human eyes and understanding what it could tell us about adaptation, about lifestyle, about how this species had survived in its world. The Denisovans, as a species, were now no longer ghost. They were real. They had faces. They had bodies. They had a geography and a timeline. They had lived alongside other human species and interbred with them. They had made contributions to the genetic makeup of modern humans that were measurable and significant. And they had vanished, leaving behind only traces in our DNA, and now, finally, the bones themselves to tell their story. By the time the 2 papers announcing the identification of the Harbin skull as a Denisovan were published, in June of 2025 in the journals Science and Cell, the reaction in the scientific community was swift. Most researchers found the evidence compelling. The combination of mitochondrial DNA evidence and protein evidence was strong. The morphological analysis, conducted in parallel with the genetic work, showed features that were consistent with Denisovans. The skull, which had been controversial for just 4 years, was now firmly identified as one of the most important fossils in the entire history of paleoanthropology. Chris Stringer, research leader in human origins at London's Natural History Museum, said that the Harbin skull was the most complete fossil of a Denisovan found so far. That statement, simple as it sounds, carried enormous weight. It meant that we now had a morphological blueprint for Denisovans. It meant that we could look at other fossils and ask whether they fit within Denisovan diversity. It meant that the entire landscape of human evolution in Asia was suddenly clearer. Not everyone was convinced. Some researchers argued that the DNA evidence, while suggestive, was not definitive enough to make the identification with absolute certainty. But the weight of evidence was overwhelming. The genetic markers were too numerous, the mitochondrial DNA too clearly aligned, the protein evidence too consistent. The debate, which had simmered for 4 years since the Homo longi designation, was largely settled. And the discovery opened up new questions. If there were Denisovans in northeast China 146,000 years ago, where were they before that? How far south did they range? The fossil record suggested that Denisovans might have lived in Southeast Asia, in tropical regions where DNA preservation was far more difficult. But if they had lived in those regions, what did they look like? What adaptations would they have developed for tropical environments? How would they have differed from the robust Northeast Asian Denisovan represented by the Harbin skull? The Harbin individual, with his large brain and robust build, suggested a lifestyle adapted to cool climates and perhaps to hunting large game. But other Denisovans might have been different. The jaw from Taiwan suggested slightly different anatomy. The handful of teeth found in various caves suggested variation within the Denisovan population. What had emerged was not a single unified species, but a geographically distributed lineage, possibly with significant variation across its range, adapted to different environments, living different lives. The implications for understanding modern human history were profound. If Denisovans and modern humans had encountered each other and interbred, then those encounters had left their mark not just in our genes, but in our adaptations. The Tibetan altitude adaptation genes came from Denisovans. Modern Asian populations carry Denisovan genetic contributions related to metabolism and immune function. The genetic legacy of Denisovans was woven through the biology of modern human populations, and now that we had a face to match the genetics, we could begin to understand more fully what that legacy meant. The discovery also highlighted the enormous power of technological innovation in science. 15 years earlier, no one could have sequenced DNA from dental calculus at this scale. 15 years earlier, protein analysis of ancient fossils was in its infancy. But by 2025, these technologies had matured to the point where they could solve mysteries that had seemed intractable. The Harbin skull had been waiting for technology to catch up to the questions that it posed. And when that technology finally arrived, it revealed its secrets. By late 2025, research teams across the world were re-examining fossils with new eyes. Any fossil from Asia that had been ambiguous in its classification was being subjected to renewed genetic analysis. The floodgates had opened. Specimens that had been locked in museum storage for decades were suddenly relevant again. Debates about species classifications that had seemed settled were being reopened. The field of paleoanthropology was undergoing a revolution driven by the combination of ancient DNA analysis, protein analysis, and classical morphological study. The Harbin skull stood as a monument to that revolution. It stood as a testament to the power of unexpected sources of genetic information. It stood as proof that mysteries that seemed unsolvable could yield to the right combination of technology, ingenuity, and scientific persistence. And it stood as a face. Finally, after 15 years of knowing the Denisovans only through fragments and genes, the world could look at what a Denisovan actually looked like. Could see the heavy brow ridge, the flat face, the massive brain case. Could understand through the morphology itself something of how this species had lived, where it had thrived, what adaptations it had made to its world. The mystery that had emerged from a pinky finger bone in 2010 had not been fully solved. Questions remained. Other Denisovans would be discovered. Other fossils would be re-examined and reclassified. The picture would continue to become clearer, more complex, more nuanced. But the breakthrough in June of 2025 was decisive. We finally had a Denisovan skull. We finally had a face. And with that face, one of the great mysteries of human evolution had moved a step closer to being understood.